Both science and art are closely linked and are ways to understand and describe the world around us. Scientists can convey knowledge through collaborations with other artists, and artists often find inspiration from science. Likewise, animation is a compelling medium for observing, communicating, and describing our world. From the complex inner workings of cells to the molecular structure of DNA, 3D animation has been an increasingly prominent visualization technique for communicating complex biological processes and behaviors. As animation can also imagine and depict possibilities not limited to the real world, can the authentic power of abstract animation provide different potentials for scientific visualization from more realistic animation? Inspired by early filmmakers and animators such as Jean Penleaf and Len Lai, who combined the scientific and poetic, I explored this question via a practice-based approach and created a short abstract animation using the motif of a virus. In making this animation, I attempted to convey 5 biological behaviors of the inner body in a creative way rather than focus solely on communicating information. The first behavior is intrusion. I used red dots to symbolize pathogens. When the virus enters a cell, it is divided into 2 parts and enters the cell to try to contaminate it. What makes abstract animation interesting from realistic animation is that it can exaggerate motion, rhythm, and color to express a sense of urgency and convey the meaning of invasion in different ways. In doing so, it can express powerful emotions, not just behavior. The process of infection is very complicated, but in abstract animation, I focused on atmosphere to transmit my idea. Based on the models and images of different viruses, I drew them with sparse lines and plans and decorated each one with bright colors. These images of the virus twist and flash on the screen, creating a disgusting and dizzy feeling. The virus replicates itself in the cell through RNA transcription. In order to enrich the animation, I kept the process of duplication, but made viruses divide into different forms. I used colorful lines and geometries to represent different viruses on a black background, which formed a sharp contrast with the soft and bright image of cells. I did this so the audience can understand which entity has a positive behavior and which is negative, almost like a character. When the cell dies, it forms lumps which push forwards and explodes. It shoots out long beaded protrusions which look like a necklace, and then breaks apart. I used red to represent pathogens and white for leukocytes. The red dot falls from the top of the frame like a death, shooting to the leukocyte and split it into 2. I based this on the actual behavior of a leukocyte which, when dying out, uses its last breath to shoot out beads in all directions to send a message. When it comes to the resistance between virus and leukocyte, the leukocyte engulfs the pathogen and digests it when receive a signal. However, in order to make the rhythm of the animation fluctuate, conflict and drama were important considerations to emphasize in animation. I preferred to give the characters personality and let the virus break through the white blood cells and confront them. Eventually, it is pulled back into the cocoon and digested by white blood cells. In conclusion, the uncertainty and mystery of abstract animation may arouse curiosity and surprise in understanding these biological processes and behaviors. Through the 5 behaviors of scientific behavior I explored, I intended to emphasize the poetry and expression of biology through abstract animation. By exploring biology as a subject in abstract animation, I wanted to show how it could demonstrate a compelling way of audiences to engage with science. Ultimately, I hope to see a greater exploration of artistic treatments of scientific subjects, and I hope my short animation is a demonstration of how abstract animation is a compelling medium to do so.